Good afternoon, everyone, or whatever time zone you may be in. Hello. My name is Nils. I'm the co-director of the University of Michigan Center for RNA and Biomedicine, and it is my absolute pleasure to welcome you to this second ever live online seminar held by the so-called RNA Collaborative. For those who are new, the RNA Collaborative came to be as one more thing that our very entrepreneurial center manager at Michigan, Martina Gerent, could do. She started a monthly online chat with the managers of a number of RNA centers around the country. When COVID-19 came around, disappointed that we could not hold our traditional annual March symposium at Michigan, the idea of holding a joint seminar series between these centers came up. And here we are, having grown by now into a collaborative of 8 centers across all of North America, including our friends in Sherbrooke, Canada. So we are international now. 2 weeks ago, the first seminar was hosted by Carla Nugebauer on behalf of the Yale Center for RNA Science and Medicine, and featured 2 fantastic talks on the regulation of RNA decapping and phase separation, as well as on the function of long non-coding RNAs in cancer. They were recorded, and I encourage anyone interested to look them up on the webpage, that's hosted currently on our webpage. We even have our own YouTube channel. The contributions from Yale are a hard act to follow, but we'll give it a try today, based on the fact that last time we ran into capacity issues with Zoom. We now have expanded into a higher capacity version of Zoom, up to 10,000 people theoretically, that could allow us to accommodate everyone who's interested in to participate across the world, potentially. The seminars will also be recorded, so you can watch them again later offline or share them with others. First up today, we have our assistant professor, Stephanie Moon. She just started this year on January, brand new. She started out her scientific career with a chemistry and biology double major at Fort Lewis College in Durango, Colorado. Moved on for her Ph.D. studies with Jeff Willish at Colorado State University on 5' to 3' RNA decay pathways, and then worked as a postdoc with Roy Parker at the University of Colorado Boulder. She is our first Center for RNA and Biomedicine faculty scholar. We hired her into the center plus the Department of Human Genetics here at the University of Michigan just this January. She is a biological science scholar at the University of Michigan, a neuroscience scholar at the University of Michigan, and has recently obtained the Brain and Behavior Research Foundation Young Investigator Award. She will today talk to us about her recent work that is centered around questions of links between translation quality control and mRNA targeting to stress granules. So without further ado, Stephanie, are you ready?
Yeah, thank you so much, Nils. Let's see, okay, cool. Got the video going and can share my screen. Alrighty. I'm very excited to be here today. Thank you so much for the opportunity. I'm looking forward to getting feedback and interacting with everybody online. So like Nils said, today I'll be talking about this new link that we are finding between the translation quality control machinery and RNA targeting to stress granules. So as you all probably know, RNA protein granules, or RNP granules as I'll call them, are these membraneless compartments in cells, and they're implicated in a lot of different biological processes. So they're coming to be recognized increasingly as these fundamental types of cellular organization mechanisms. And they're involved in processes as diverse as germ cell organization and neuronal biology. And what I'm interested in mostly are how stress granules and P-bodies might play a role in regulating translation and RNA stability, especially during stress, context, and disease. So what we'll be talking about a lot is how RNAs are regulated during this process called the integrated stress response. And the integrated stress response is this really important conserved eukaryotic pathway that helps cells adapt to and respond appropriately to a lot of different stress conditions. This includes heat stress, nutrient deprivation stress, and also arsenic stress, which we use a lot in the lab. It's a really robust way to cause an integrated stress response. So all these different stressors will activate EIF2 kinases, and these phosphorylate a major regulator of translation initiation to globally suppress translation activity and end up reprogramming gene expression. And as soon as translation is suppressed, you end up forming stress granules. So what I was curious about when I started my postdoc in Roy's lab is how RNAs are coordinately regulated during stress. So we know that translation is rapidly shut off, as I mentioned, during the stress response, and you can see that through bulk labeling of messenger RNA or nascent proteins during stress. And then at the same time, mRNAs are localized to these granules called stress granules, and you can see these through oligo-DT fish staining. But what we weren't really able to do at the time was see how RNAs were regulated at the levels of translation and localization to RNP granules at the same time. So it was hard to say exactly how these two processes were related to one another. So I really wanted to understand kind of what the causes and consequences of the localization of mRNAs to stress granules were in terms of how those RNAs were functioning. And we ended up finding a bunch of different things regarding how RNAs interact with stress granules through a collaborative project that I'm going to mention today. And that work led to our newer discoveries of how translation quality control factors might play a role in this whole process. So today I'm going to talk about three different things in this talk, and we're going to start with kind of the fundamental properties of RNA stress granule interactions and end with how we think the translation quality control factors might be involved in this process. So first I'll talk about our past work on fundamental properties of RNA stress granule interactions. So this project really came about because I saw recent work from Dr. Tim Stasevich's lab at Colorado State University. They developed this method called nascent chain tracking, and this allows you to visualize single mRNAs in living human cells. You can see their translation status and the localization of the RNAs. So in this movie, you can see these little RNA foci in red, and the nascent chains, protein chains in green. When they co-localize, you get little yellow dots. And so the way this works is you can put a plasmid into cells that has a repeat epitope tag at the N-terminus of the nascent protein that's visualized by an antibody fragment that you introduce into the cells. And then the mRNA itself is labeled with an MS2 coat protein. So you can simultaneously track the mRNA's localization and the nascent protein chains that are indicative of nascent translation. So we decided to try and use this method together to figure out how RNAs interact with stress granules in terms of their localizations and their translation status. Because it was widely assumed that mRNAs couldn't interact with stress granules when they were translating, but there's technical limitations with actually observing that. So when we introduce these constructs into cells that have a green fluorescent protein that marks stress granules, which I'm showing in blue in these images, you can see that in this unstressed cell, there's no stress granules, but you have lots of little mRNAs, and they are translated or not translating depending on the presence of these green nascent proteins. But in stress cells, you see there really isn't much translation happening, and mRNAs start to actually accumulate in stress granules. So you can see the stress granules are these green or blue blobs here, and then the mRNAs are in red. So the first thing I noticed when I started going through and trying to analyze how RNAs interact with stress granules is that RNAs could interact very stably with stress granules and be sequestered within them, which was kind of another assumption that a lot of people in the field have made over time, is that if an RNA went to a stress granule, it was stably sequestered within that stress granule. And that's important because if RNAs can't translate inside of a granule, then the presence of the RNA in the granule indicates that that RNA is translationally suppressed during stress. However, a lot more frequently, I saw interactions of RNAs with stress granules that were highly dynamic. So we could see RNAs kind of bumping into stress granules, interacting with them, co-moving with them for several seconds, and then leaving again. And what Tim and Tatsuya were able to do is actually extract all of this information from many, many movies that we took. And they were able to create a model for how RNAs actually interact with stress granules in kind of a high throughput and analysis pipeline. And what they found kind of mirrored what I had seen by eye, and that is that mRNAs can interact in a very dynamic or transient state with stress granules, but they could also interact in a very stable kind of sequestration mode with stress granules. So it turns out there's two ways that RNAs can interact with stress granules. And we found that there was three things that really governed how an RNA interacted with the stress granule. The first thing was if an RNA was translating, it rarely, if ever, engaged in a stable sequestered interaction mode. The second thing is that the larger the stress granule was, the more stably an mRNA could interact with it. And vice versa, the same thing was true. The longer an mRNA's open reading frame was, the longer that RNA could interact with the stress granule. So this study was published last year, and we basically described some of these fundamental parameters for how RNAs interact with stress granules. They can interact very transiently with them or be stably sequestered within them. The same thing was found for P-bodies as well. And then we found that higher valency kind of corresponded or correlated with the duration of these interactions. And most importantly, which will be good to remember for the next part of this talk, translating RNAs did not become sequestered within stress granules, but rather they docked transiently with stress granules. So that brings me to the next part of the talk about how we found translation quality control factors are involved in mRNA trafficking to stress granules. So we were interested in looking at how translation quality control machinery could impact RNA regulation in stress granules during stress, because a lot of these translation quality control factors are implicated in neurodegenerative and neuromuscular diseases. One of those factors is called velocin-containing protein, or VCP as I'll call it. So VCP is known as a ubiquitin segregase, and what it does is it latches onto ubiquitinated proteins that are targeted for degradation, and it helps pull them out of protein complexes to allow them to be degraded. And we knew from past work from Roy's lab and from other labs as well that VCP is required for proper stress granule disassembly. The mechanism for how this happens isn't clear, but it does seem to be important for actually disassembling stress granules when stress is resolved. So we decided to try and use a single mRNA imaging technique in collaboration with the Stasevich lab to figure out if VCP could actually be, removing RNA protein complexes from stress granules to help destabilize stress granules. And our hypothesis was really that VCP would be playing a primary role in the recovery from stress, not in any other stage. So what we did is just inhibited VCP during stress using this single mRNA imaging approach. And we expected that mRNAs might be sequestered in stress granules for longer if it was playing a major role in stress granule disassembly through this mechanism. But instead we saw something really surprising. And that is that when we inhibited VCP, there was lots of mRNAs that appeared to still be translating during stress. And the same thing was true for proteasome inhibition. So if you inhibit the proteasome, which has also been shown to be important for stress granule disassembly, you get a lot more nascent proteins in cells that are stressed with arsenic than when you just stress them alone. We also found that, as was previously shown in our past manuscript, that these translating RNAs still transiently interact with stress granules. But the next question was, is translation actually still being suppressed during stress, or could ribosomes potentially be stalling in this context? Because there's really only two ways that we would see more nascent proteins associated with mRNAs in stress conditions when you inhibit VCP or the proteasome. And it turns out when you do a Harringtonian assay, which can inhibit the very, very early stages of elongation during arsenite stress, that ribosomes appear to stall or pause on these reporter mRNAs during stress when you inhibit VCP. We also did some metabolic labeling experiments and polysome gradients and found that translation is still globally suppressed in these contexts. But on some mRNAs, it turns out that the nascent proteins are still associated with mRNAs despite there being robust translation shut-off. So this brings us to an interesting kind of observation, and that is that in the context of global translation initiation shut-off, there appears to be some mechanism where translation elongation is suppressed. And this inhibits ribosome runoff to some extent, but in normal conditions, you should have VCP and proteasome activity there to help potentially remove ribosomes and allow mRNAs to exit translation. So this observation actually makes some sense in the field because there are other papers that have used different approaches, such as ribosome profiling. And they found that in yeast and mouse cells, for example, a lot of different stresses cause ribosomes to accumulate in mRNA open reading frames, suggestive of ribosome stalling. So the question is now, how is VCP and the proteasome actually engaging in this process, and does this have an impact on mRNA localization to stress granules? So to try and address this question, we used an analysis called single molecule fish, single mRNA fish. So what we had observed before is that VCP and the proteasome seem to be important for removing mRNAs from translation. And the idea now is that if RNAs are trapped in translating stalled complexes, the mRNA won't be able to go into the stress granule. So we used single molecule fish to try and see if there's an impact on mRNA localization to stress granules. And in this way, we could detect endogenous mRNAs and not rely on reporter RNAs exclusively. So we're looking at fixed cells and whether or not RNAs are localized to stress granules. And importantly, we can use a translation inhibitor called puromycin, which will force mRNAs to be released from ribosomes, whether they're stalled or not. And in this way, we can kind of control for changes in translation. So the first observation we made is that when you stress cells with arsenic and you add VCP inhibitors or proteasome inhibitors, this representative mRNA called ANAC in the cell is significantly reduced in its localization to stress granules. When you treat these cells with puromycin, they're rescued in their localization to stress granules. And the same is true when you treat cells treated with proteasome inhibitors with puromycin. You get more mRNAs localized to stress granules. And so this told us that rather than there being sort of a general impact on stress granule biology due to proteasome or VCP inhibitor treatments, that these adding puromycin can rescue these defects, which directly links this process to the transition pathway. And in addition to the puromycin control, we can see that the NORAD long non-coding RNA is unaffected in its localization to stress granules in these conditions. And furthermore, in heat stress condition, we also see a significant reduction in ANAC recruitment to stress granules when we inhibit VCP that's rescued with puromycin. And NORAD RNA again is unaffected, but in ER stress, there is no change in RNA localization, whether or not VCP inhibitors are present. And so that tells us that this pathway is important for certain stresses, but not all stresses. So when we observed that mRNAs seem to be stalled in translation elongation during stress, when we inhibited VCP or the proteasome, these mRNAs are unable to now go and interact with stress granules. So it does seem like there's an important role for VCP in the proteasome in mediating RNA recruitment to stress granules. So we wondered how this could actually happen. And VCP and the proteasome as well, they're known to be part of this RNA ribosome-associated quality control pathway, which is an important surveillance mechanism that exists to really destroy aberrant mRNAs and proteins before they can be made and create toxic or aggregation-prone products. And the context where the RQC pathway is important are in conditions where mRNAs have large stem loops or internal poly-A tracks or are truncated open reading frames, for example. And this pathway is thought to result in the degradation of nascent proteins and ultimately the degradation of the mRNA as well through no-go decay and other RNA decay pathways. So we wondered if other components of the RQC pathway could be involved in this mRNA release from translation during stress that we were observing. So in this experiment, I knocked down LTN1 and NEMF, which are key components of the RQC pathway. And I found that indeed they were required for full mRNA recruitment to stress granules. When you treated cells with puromycin, you can actually rescue RNA recruitment to stress granules. So that places LTN1 and NEMF in the same type of pathway downstream of translation as VCP and the proteasome. And we also took kind of a genetic approach to see whether or not VCP and the proteasome were acting in the same pathway as LTN1 or NEMF, or if they were acting in parallel pathways. And we found that there was no increase in the reduction of mRNA recruitment to stress granules when you inhibited both VCP and LTN1 or NEMF, and the same was true for the proteasome. So it turns out that the LTN1, NEMF, VCP, and the proteasome seem to all be important for removing mRNAs from stalled ribosome complexes in the same pathway. They're acting in the same pathway together. So to kind of try and flesh out the events of this pathway a little bit more and see how similar to the canonical RQC pathway our stress-activated RQC pathway is, we decided to test how a reporter RNA that has an internal poly-A tract that was developed by the Ph.D. lab would actually be affected in stress. And so we used this reporter construct that has internal poly-A tracts, which is called the K20 construct, because it makes 20 lysines, or a K0 control reporter RNA that had no stalling sequence in it. And we found that, yes, this reporter RNA is acting like it's stalling because we can measure the GFP to RFP ratio from this construct, and did a fish experiment to detect how this RNA was affected during stress. And what we found is that while this control RNA transcript that doesn't have a stalling sequence in it does require VCP activity to localize fully to stress granules during arsenite stress, this K20 reporter RNA that does have a stalling sequence in it is unaffected by VCP inhibition in its localization to stress granules. So this observation suggests that the stress-activated RQC pathway that we were studying is substantially different from the canonical RQC pathway, even though some of the same factors are involved in both pathways. So in the canonical RQC pathway, very specific RNA substrates are targeted, whereas in our stress-activated RQC pathway, apparently kind of normal constitutively expressed RNAs are targeted. We also found that the order of events is different for the canonical RQC. So in this pathway, other labs have shown that ribosome splitting precedes RNA release and indeed VCP activity on the nascent peptides. But in our assay, we're finding that when you inhibit VCP or the proteasome, nascent chain ribosome complexes are still present in the cell. So that suggests that VCP and the proteasome are acting before mRNAs are released from ribosomes. And then finally, the fate of mRNAs seems to be substantially different. So mRNAs are degraded in the canonical RQC pathway, but in the stress-activated RQC pathway, they can localize to stress granules. So there's some implications of this SARQC pathway, as we're calling it. That is that ribosome stalling does occur in a lot of different stress conditions, and that a mechanism may exist that involves VCP that resolves certain ribosome pauses without destroying mRNAs and nascent proteins. Also, stress granules could be storage sites for these mRNAs that are stalling in translation. Perhaps they're aberrant in some way that remains to be detected. And finally, there's several disease contexts that could result from defects in proteostasis, and maybe the SARQC is involved. So for example, in a preliminary analysis, we found that overexpressing pathogenic VCP alleles actually increases mRNA recruitment to stress granules. But the full implications of this still need to be tested. I'll be working on that in the future. So with that, I'd like to thank everyone at the Parker Lab, especially those who directly contributed to this project, and Roy. In addition, our collaborators, Tim Stasevich, Tati Morsaki, and Ken at Colorado State University, our funding sources, and then everyone at the University of Michigan. I just started my lab in January, and I'm really looking forward to following up on this research. And with that, I will take any questions.
Very nice, Stephanie. Thank you very much.
Thank you.
I think there are a couple of questions. I first wanted to also say I appreciate certainly that you used the Michigan cap for the 5' cap. I appreciate that. So very well done. And of course, it's great to see how phase separation can regulate all sorts of processes in the cell upon outside cues, basically. So there was one question that got a couple likes, I guess, and boosts about how exactly you tag the stress-related mRNAs for your studies.
Yeah, so we used 3 different methods to detect RNAs in these studies. The first method is called nascent chain tracking. So the method developed in Tim Stasevich's lab, really. So basically we use MS2 stem loops in the 3' untranslated region of reporter RNAs. And we use 3 different open reading frames of nuclear proteins because those help with reduce the background for imaging in the cell if you create nuclear proteins. The nascent proteins are detected by introducing antibody fragments that are fluorescently labeled into the cell. So we use halo MS2 constructs to actually detect the mRNA. The second method that we used was called the SUNTAG method. So we used cell lines that were developed in another lab, and those are genetically encoded SUNTAG proteins, but I didn't share that data today. And then the third method was just using single molecule FISH, so you just use probes to detect endogenous mRNAs. Lots of different tools, all used whenever needed.
Carla Nugebauer had this question, also got a couple likes. The significance of docking of the mRNA on the surface of stress granules specifically.
So we don't really know how long an RNA has to interact with a stress granule to be affected by it. So the docking interactions were, I think, on average about 40 seconds for one of our reporter RNAs. So that's kind of a long time if you think about, you know, for example, the interaction rates of certain RNA binding proteins with mRNAs in the cell. We don't know if the docking has an effect on RNAs, but it's possible that the composition of an RNP could change after a transient interaction with the stress granule.
Another question that has come up multiple times, whether you looked at M6A modified mRNAs and their relationship to stress granules.
That's a really great idea. It's something I'd really like to do in the future, but I think that those experiments need to be done really carefully, and but now we should have the technology to actually address those questions, but I have not done that myself yet.
And Tatsuaki Kurosaki asks that G3BP also binds to structured mRNAs, and the question then becomes whether there's evidence that structured mRNAs like G-Sig rich have a preference to form stress granules.
That's a good idea. I don't know if there's very strong evidence that RNA GC content influences the localization of an RNA to stress granules, but I can imagine a lot of cases where structured RNAs could be differentially enriched or de-enriched in stress granules. But I have not specifically looked at that. I know there are several transcriptome papers looking at the composition of stress granules, and then there's other really recent papers about G3BP and structured RNAs, but I haven't delved into those myself.
And another question is whether poly A binding protein, PAP1, binds K20 and could interfere with exactly the model or the interpretations of the model?
Okay, that's a good idea. So the question is kind of about why is that internal poly A tract RNA unaffected by VCP inhibition?
Exactly.
I think I really believe that what the implication of that is that the RNA is being degraded, and if it's translated, it's degraded. So once the ribosome stalls, the RNA is degraded, and then VCP acts on the nascent proteins. So I think VCP is acting downstream of where the RNA is released from the ribosome. They're being endonucleolytically cleaved. But it is possible that PAB or another protein is binding that poly A tract. The thing is, I would expect all the mRNAs in the cell have PAB associated with their poly A tract, so I'm not sure if PAB's presence or absence would affect the SAR-QC pathway, but yeah.
Lots of other questions that are popping up and moving up and down, I think, in the popularity. So I'll just take one more. Here, that was about how mRNAs escape translational repression by eukaryotic IF2 alpha phosphorylation.
So I think what we're seeing in arsenite stress are this subset of mRNAs that are translationally repressed at the initiation phase, but not during, but the ribosomes are not running off. So I think the reason we're seeing nascent proteins on these mRNAs during arsenite stress is not because they're continuing to undergo translation initiation, but rather because the ribosomes are stalled and they're not falling off. But these RNAs do undergo translation suppression at the initiation stage. That was something that we had shown previously as well.
And there are a couple other questions in the Q&A session that you can maybe look at offline.
Sure.
All right, thank you very much again for a great presentation. Again, anyone who wants to still ask questions to Stephanie could put them into Q&A for her. Excellent presentation. So now we are off to our second presenter of today. And I think the idea of today's presenters was to kind of give you a spectrum of different things that people can do in the RNA field, broadly defined, of course. And so Peter Freddolino is essentially really the opposite. He works on bacteria and he works with computational tools primarily. So the opposite of what you just saw. He started out in 2015 here at Michigan with an NIH Pathway to Independence Award. And before that, first worked got his undergrad degree in biology at Caltech, and then worked in University of Illinois Urbana-Champaign with the late Klaus Schulte in biophysics and computational biology. And this was really a very computationally heavy work that he then followed up with a postdoc at Columbia University and Princeton University, in the other order, Princeton, then Columbia. And that led him to us now to work on bacteria and the question of large-scale chromosomal structure in bacterial transcription. Peter, take it away.
Well, thank you very much, Nils, for the introduction, and thank you all for being here. So I'm going to zoom to the exact opposite end, in a sense, of the RNA life cycle and just talk about how transcriptional decisions are made in bacteria. And really want to highlight some interesting findings we've had recently on the role of chromosomal structure and some almost heterochromatin-like domains of the bacterial chromosome on the regulation of transcription. But I want to take a step back to give a little background on how we got to this in the first place. So my interest in this general area got started with meta-analysis that I had done when I was a postdoc, where we collected knockout fitness profiling data in E. coli from many different sets of studies to look at how often loss of function of different genes could be either beneficial or deleterious to the bacteria. In this case, it was done in E. coli. And what we found was under almost any condition that one tried, there would be several single gene knockouts that were actually beneficial, that improved fitness. And so what I'm showing here is the y-axis is a log 2 of the number of significant null mutations that were either beneficial in blue or deleterious in red to the cells. Yeah, sorry?
I'm not sure, but you are not sharing your screen. Is that possible?
Oh, wow, fantastic. Thank you, Nils. That would have been smart, wouldn't it? All right. Wow. Okay, can we see a graph now?
Perfect.
Excellent. My apologies to everybody. So now that you can actually see the graph, what we've got here on the y-axis is the number of null mutations that either had a significant positive in blue or negative in red effect under each of 144 different non-redundant conditions that we looked at. And what we found really interesting was that under all but 5 of these conditions, there is at least one beneficial null mutation. That is a knockout where the cells with that knockout do better than the wild type. And when we dig a little deeper, we can look at different classes of genes and look at which ones are overrepresented among those beneficial nulls. And what we see as the most overrepresented classes are enzymes, probably not surprising, and then regulators. And so what we get out of this are a couple, or of the study in general, were a couple of important design principles for bacteria. Number one, that their biochemical capabilities often exceed their regulatory capabilities. And I say this because we can so readily find cases where if the cells just did not express a particular gene under some stress condition, they would actually do better, since to a first approximation, those single gene knockouts are going to be similar to just not expressing those genes. Another aspect of this was the finding that poorly annotated proteins still showed up at comparable rates to everything else in terms of their fitness effects. So they're also important. And so to get a better ability to both understand and eventually design bacterial transcriptional regulatory networks and their responses to the environment, we really need to get a comprehensive profile about what they're expressing at a given time. That part's easy. And also why, the molecular logic. And so that led us into a methodological question. How can we even do that? And the techniques that are typically available don't actually meet all the criteria that we would have. We could go do RNA-seq experiments, which is the first thing you'd think about if you want to see what is getting transcribed, but that doesn't tell us the why, the molecular logic that is driving the transcriptional regulatory pattern. And we can do a bunch of ChIP-seq experiments, which will tell us where one protein is bound, but E. coli has more than 200 transcription factors. We're probably interested in multiple different conditions, and this leads us to a really intractable combinatorial explosion of experiments that you might have to do. And so that leaves us then without a good method for figuring out what is actually driving a complex transcriptional regulatory response. And I'll just say as an aside here, that just to make life more difficult, about a quarter of the transcriptional regulators in E. coli have very little information known about them. So we don't really have all of the information that we would need to do ChIP-seq on all those transcription factors, even if we had armies and armies of grad students to go do them. So the approach we take instead is a method that I had worked on while I was a postdoc called in vivo protein occupancy display at high resolution. So the idea behind this method is instead of measuring the occupancy of different transcription factors one at a time, we want to get a single one-dimensional profile of where all the protein occupancy is on the bacterial chromosome under a physiological condition of interest. So the way this works, we grow the cells under some physiological condition that we're interested in, cross-link protein to DNA, and then we lyse the cells, enzymatically shred the DNA, and up until this point it looks like a ChIP-seq experiment. But instead, once we have this digested soup of protein DNA complexes and free protein and free DNA, we throw this into a phenol chloroform extraction. And what several people have shown is that you'll get an enrichment at the interface between the aqueous and organic layer for the cross-linked protein DNA complexes. So we can recover the DNA from this interface layer, sequence it, and compare it to an input sample, and then we'll get this one-dimensional readout for where there is protein bound under physiological conditions. And so this gives us then a profile that we can deconvolute into the contributions from all the different regulatory proteins that we might be interested in. So with this method in hand, it's useful first to just take a look at an area where we already know the right answer and see what our method tells us about the regulatory logic there. And so right here, I'm showing my favorite promoter in E. coli, or one of them at least. This is the promoter upstream of the gene PureC. Now we know that PureC expression goes up if there are no purines present in the media, and we also know why that is. There is a binding site right here for the purine repressor PureR. That will be bound and repressing transcription if there are purines present in the media, and that binding will go away if there are no purines present in the media. And so if we look at the total protein occupancy profiles that we can measure with our iPod-HR method, in wild-type cells in rich media, you get a nice occupancy peak right here, right where that PureR binding site is supposed to be. So far, so good. If we shift the cells into minimal media where there are no purines, we see that peak go away. And likewise, if we keep the cells in rich media but delete the purine repressor PureR, that peak goes away. So that's lovely in that we were able to recover what we already knew about the regulatory logic here. But the nice thing about this method is we did not have to decide ahead of time that we were looking only at PureR occupancy, and we didn't have to decide ahead of time that we were looking at this regulatory region. We get this information across the entire chromosome under each and for all of the different proteins that we are able to detect with this method. So I'm going to fly through some interesting things we can get when looking at the occupancy of individual transcription factor binding sites with this method. There's a paper in bioRxiv now that goes into all of these in detail, but some of the things we can do is look at the dynamics of the binding of a bunch of different transcription factors across physiological conditions. So for example, here I've got a bunch of different regulators along the X here and different conditions along the Y-axis, and we can see how active they are under different conditions. We can identify what proteins bind to newly identified sites and de-orphanize the regulatory logic of some genes because we're able to see where the binding site is and when proteins bind or don't bind to that location. And we can also identify motifs that are enriched in the highly protein occupied regions, in the things that look like transcription factor binding sites. Some of these correspond to known transcription factors, and others probably correspond to those currently poorly annotated ones because we get equally strong motifs that don't match any known transcription factor binding sites. But what's really, I think, the most interesting thing to come out of this comes if we zoom out a little bit and instead look at the chromosome over length scales of a few kilobases. And so that's what I'm showing now. Please note the scale here. This is a little chunk of chromosome, about 50 kilobases. And what I'm showing here is the total protein occupancy in blue and the total amount of transcript coming from these regions of the chromosome in orange. And so there are two really interesting things that we see here. Number one is that on the E. coli chromosome, there are a few hundred regions that are just walls of protein occupancy over scales of kilobases or more. And we refer to these as extended protein occupancy domains. So they're just large, highly protein occupied regions on the chromosome. And the other thing that's really striking is if we look at the amount of transcript being produced from those regions, there's a very strong anti-correlation at this length scale between the total protein occupancy and the amount of RNA that's showing up from these regions. So the extended protein occupancy domains tend to have very little transcription going on, and then there are other regions that are highly transcribed that don't have these walls of protein occupancy on them. So we can do a little bit of just bioinformatic analysis of what the sequence features are of the regions that are in these extended protein occupancy domains. And a few things that pop out readily is they tend to be AT-rich DNA. They tend to be highly flexible. They are usually transcriptionally silent across the entire chromosome. And there are enrichments for binding sites, both known and predicted, for the nucleoid-associated proteins of E. coli. So this is the closest analog that E. coli has to histones. They tend to be structural proteins. And then another interesting thing we can find is if we look for classes of genes that are enriched or depleted among the extended protein occupancy domains, we can find several Go terms that are enriched in the EPODs. And so any Go term that is red in this column is enriched in EPODs. Anything red in this column on the left over here is depleted in the EPODs. And so two categories that particularly stand out to us as enriched in these extended protein occupancy domains are transposons and the cytolysis Go term. That is typically indicative of the presence of a prophage, an integrated viral genome, because many of those prophages actually still have lytic operons that can actually cause the cell to lyse if they happen to become active. So at this point, we know there are these large, highly protein occupied, transcriptionally silent regions on the chromosome. And that brings us to what I call my chicken and egg question, which is, do these highly protein occupied regions just form where there is no transcription occurring, or are they actually silencing domains on the bacterial chromosome? And so to answer this question, we made a fun little reporter that we then wanted to integrate randomly into the chromosome to see how well it was transcribed, depending only on where it was. And so this is a uniform reporter where we have strong bidirectional terminators on either end. We have a curable antibiotic resistance marker, and then we have an inducible promoter, little fluorescent protein. And then really important, we have a unique molecular barcode on every molecule of this, so we will be able to associate the transcript that comes off of this thing with the location on the chromosome that the corresponding sequence had integrated into. So then we can use TN5 transposase to randomly integrate this into the E. coli chromosome, and then we can induce transcription, purify the RNA and DNA, sequence them separately, and look at the counts of each barcode in the RNA normalized by the amount of DNA present from the same barcode. And what that tells us is what we call the transcriptional propensity, which is controlling for DNA abundance, how transcribable that reporter was based on where it integrated into the chromosome. And we're able to get this for 144,000 different trackable integration locations on the chromosome. And so when we look at the profile that we get out of this, this is what the profile looks like. The y-axis here is the transcriptional propensity along the length of the chromosome. And we can see there's a fairly broad range of effects coming solely from location, where we have very highly transcribable locations. There are the peaks here and here and here, for example. And then on the other hand, we have some very low transcription regions as well. And just as a quick answer to my chicken and egg question earlier, as I will show in a couple slides, these low transcription regions line up very nicely with the extended protein occupancy domains that I was showing a few slides ago. Now, just to start to get a feel for what the purpose might be of these variations in transcribability, we can look at classes of genes that are either enriched in the high transcriptional propensity regions or the low transcriptional propensity regions. And in the high propensity regions, this is just a sampling. These are not all of the terms, but enriched in the high transcriptional propensity regions, we see things like the ribosomal RNAs and also ribosomal proteins, and also things involved in amino acid biosynthesis. And then on the other hand, in the low transcriptional propensity regions, we have enrichments for the same things we saw in the extended protein occupancy domains, things like prophage components and also transposases. So it looks like highly consistent with what we were seeing from this picture of the extended protein occupancy domains. The bacterial chromosome has these large, highly protein occupied regions, and they tend to be transcriptionally silencing both to the genes that are already there and anything new that we put into them. And so if we look, for example, just at the profiles of transcriptional propensity inside of EPODs versus everywhere else, we can see this lower transcriptional propensity inside of EPODs. Now, at this point, we started to wonder, what are the molecular features that are actually forming EPODs? And so the way we started to approach this was to do individual knockouts of nucleoid-associated proteins. And what we saw pretty quickly was that the general rule seems to be that these extended protein occupancy domains tend to be composed of a mix of about 2 different nucleoid-associated proteins. And we were able to find several classes of them, several classes of pairs of nucleoid-associated proteins that give rise to these regions. So just to give one example of what this looks like, Here is one extended protein occupancy domain. It's on the WA operon in E. coli. And so you can see this nice wall of protein occupancy in wild type cells. If we delete the nucleoid-associated protein HNS, we see a quantitative reduction in the occupancy in this region, but we still have the wall of occupancy present. If we delete a different nucleoid-associated protein, STPA, we also see occupancy that looks basically like wild type. But if we delete both of them at the same time, we see a dissipation and a fundamental change in the way the protein occupancy looks here. And so this is all consistent with what others have shown about the particular interplay of HNS and STPA, is that HNS tends to form filaments on the chromosome and silence regions of the chromosome. It also represses STPA, but STPA can act as a partial fill-in in the absence of HNS. But what we've been able to do is, starting from the protein occupancy profiles we get from all of the single mutants, we've been able to identify now some other classes of extended protein occupancy domains that carry out other biological functions. It looks like I'm running a little short on time, so I may skip the tail end of this and go a little fast through it. But I think the most interesting case we've looked at is the case of the interaction between the proteins FIS and HFQ. If we looked at the effect of single mutations of FIS and HFQ on accessibility to transcription, we saw across the chromosome that they were very highly correlated with each other. So we thought, okay, it makes a lot of sense then. Let's go make the double mutant of FIS and HFQ. Turned out that we simply could not make that double mutant. It looked like there was a synthetic lethality. And so to give an idea of what this looks like, this is the efficiency with which we are able to either make a HFQ FIS double mutant. This is the orange bar over here, compared with just deleting HFQ in wild type cells. And here's a control transduction. So we can see that there's a little bit of a drop in the efficiency of putting knockouts into the delta FIS background for any gene, but you basically get no colonies when you try to delete HFQ in the delta FIS background. And the one or two that you do get die when you restreak them. So you really can't make this double mutant. So to figure out what was going on here, we went back to our categories of genes that were enriched or depleted in the extended protein occupancy domain, since we saw that there were a bunch of extended protein occupancy domains that lost occupancy when you deleted either one of these factors individually. And so reminded of the fact that there were transposons and mobile elements enriched in these domains, we went and tried to do the same experiment in a genetic background called MDS42, where all of the prophages and mobile elements had been deleted. And sure enough, when you do this in MDS42 cells, you're able to make that double mutant of FIS and HFQ with the same efficiency as our control transductions. So something that was deleted in one of the MDS42 regions apparently is what's causing the synthetic lethality of those two deletions. So to identify what that might be, we went looking for extended protein occupancy domains that lost occupancy in either the FIS or HFQ single mutants. And this is an example where we have wild type occupancy in blue. You lose some occupancy with either the FIS or the HFQ single. And then we thought that these might be candidates for things that are causing the cells to die because they're being de-repressed when you delete both FIS and HFQ. So to test this, we identified 3 of these regions that we call R1, R2, and R3, and tried making the FIS-HFQ double knockout in the presence of deletions of any of those regions. And what we saw is that deletion of either R1 or R3 was sufficient to restore the ability to make the FIS-HFQ double knockout. And we have some more recent data I don't have shown here that shows if you delete both R1 and R3, it looks essentially like the MDS42 numbers. And so what this shows us is that there is this extra class of extended protein occupancy domains that's dependent on FIS and HFQ acting together, and that is absolutely essential to silence integrated prophages, which are otherwise de-repressed and kill the cells. And we've actually at this point been able to identify 3 different classes of extended protein occupancy domains that seem to play different physiological roles and are repressed by different pairs of proteins. And one last quick point is that we've been doing these experiments now in many other bacterial species, and we've seen similar broad patterns of protein occupancy in every species we've looked at. Just for example, here is a set of profiles from Bacillus subtilis, where we look at RNA polymerase occupancy via ChIP-seq on the top 2 tracks. total protein occupancy by iPod on the bottom two tracks. We can see most of the chromosome appears to be open, but there are large regions that correspond to our definitions of extended protein occupancy domains that are highly protein occupied and transcriptionally silent. And we see the same kind of patterns in Vibrio cholera and Caulobacter crescentus so far. And so at this point, unfortunately, I'd have to skip through a little bit more material where we've looked in more detail at what defines regions of high and low transcriptional propensity for the sake of time. But we have papers, there's one in Cell Systems covering the aspects of what defines high and low propensity regions, and one that was in Cell earlier this year discussing the contributions of three-dimensional chromosomal structure to the regulation of genes in E. coli. And so just to let people actually have time to discuss or to ask questions, I'll skip ahead to my acknowledgements and thank all of the folks in my lab and the collaborators that we had worked with on this. And I'm happy to take any questions that people have, which I think Nils is going to be handling through the Q&A interface.
Yeah, excellent. Thank you very much, Peter. Great presentation. It's nice to see that not only CRISPR parts of genomes of phages get incorporated into bacterial genomes, but other parts also that actually are full phages that can go somewhere and have to be repressed.
And yeah, and there's definitely some fun defense mechanisms that we're just now getting at that also seem to play a role. Absolutely.
Mm-hmm. All right, so we already got a couple questions in the Q&A window. One of them is, and that seems to be a popular one, is how dynamic are these domains upon prophage induction?
Okay, so that's an interesting question about prophage induction. That is not one that we've looked at yet. I'll say a couple different things about that, because I think it's a great question. It's a great series of things we're interested in. So the question of how dynamic these things are in general, it seems to be not very. Under all of the different physiological stress conditions that we've looked at several different genetic perturbations, nutrient starvation, things like that. There are no more than about 10% of these things that move around. Now, the question of phage induction in particular, we haven't tried to induce any of the phages to track occupancy. What we have looked at is sort of the opposite, where we've been working with Ursula Jacobs' lab here also at Michigan to look at what happens if we weaken the extended protein occupancy domains by deleting one of the pair of proteins that forms a set of EPODs that are on prophages. And we see that actually makes the cells more vulnerable to phage induction. So the phages will get induced by lower levels of DNA damage and end up killing the cells much more readily. So it looks like they're playing a strong silencing role, but we haven't actually been tracking what's happening to the occupancy on those regions when the phages are being induced. We just know that it de-represses everything.
Yeah, one question that kind of, I think, channels over to eukaryotes a little bit is whether these EPODs silence other mobile elements beyond prophages. I mean, and this might be applicable to higher eukaryotes perhaps, but introns, transposases, and other things.
Yeah, well, so I mean, obviously I think that this is usually the domain of things that are being silenced by heterochromatization in a lot of eukaryotic systems. And currently, so we know that transposases in E. coli are among the things that are being silenced by these EPODs. And we're drawing a lot of inspiration right now from especially plant biology in terms of trying to figure out how those EPODs form. And the reason we're doing that is because HFQ seems to play an instrumental role in forming the EPODs. And it is both a RNA binding protein, where it's better known, and a DNA binding protein. So exploring some sorts of transcription dependent mechanisms that may be involved in establishing silencing. That would certainly have analogies to transcription dependent silencing that happens in plants. But we're really in the early stages of that right now.
And related to that, perhaps, are epigenetic phenomena playing a role in these EPODs? So maybe chemical modifications or others?
Yeah, that's also a good question. And this is also something we're really deeply looking into. So the best characterized DNA modification in E. coli is going to be DAM methylation, but that's theoretically everywhere. And no one has found any strong differences in the DAM modification rates in different places on the chromosome. we highly suspect that there has to be something modified on either the DNA or the protein in the EPODs to define where they are, because we have not found a strong sequence signature of what is going to make an EPOD. So that's something we're currently looking into by trying to specifically pull out the proteins from EPODs and do some mass spec and see what modifications are on them. We know that there are post-translational modifications on the E. coli nucleoid-associated proteins, but we don't know yet how clustered they are on the chromosome. So I think we're somewhat behind the state of knowledge in eukaryotes, but I suspect that there's something analogous to a histone code going on here that's going to help define the silenced regions.
All right, and I'll take one more question because we are running slightly after the hour, so but we started 2 minutes late. Do you think that EPODs also could potentially be involved in gene activation, so the opposite? Well,
so that's a great question, and I think there's a little bit of subtlety about what one calls an EPOD. There was an original paper on these methods that brought up the idea of EPODs. This is from my postdoctoral lab, where there were two classes of EPODs, one that was called highly expressed EPODs and one that were transcriptionally silent EPODs. And so these were both regions of high protein occupancy. One set was getting transcribed, one wasn't. And it turns out from the follow-up studies we've done, it's clear that what were called highly expressed EPODs were really just regions of high RNA polymerase occupancy, because RNA polymerase is, shockingly enough, a protein. It shows up in these occupancy profiles. And so we now subtract off the RNA polymerase occupancy by doing an RNA polymerase ChIP-seq and removing that signal from the total protein occupancy in order to get what is effectively everything else. Now, I don't know if that gets directly at the question, which is, do these things play a role in activation? We certainly have not found any case yet where we see something getting activated by a large protein occupancy region like this. That tends to be much more the domain of individual transcriptional activator binding sites. That doesn't mean it doesn't happen, but it's not something we've seen yet. It's certainly something we'll keep our eyes open for, and it's a great question.
There was one other question actually I wanted to pick up, was early on, whether your technique also picks up proteins that are not directly associated with the DNA, but in a larger complex through other mediators.
Yeah, so that is going to be entirely dependent on the cross-linking that one uses. And so because our readout is by DNA sequencing, we're not directly looking at the proteins at all. We have mass spec'd the stuff we pull out of that, and it looks like we do also pick up other proteins that are indirectly associated when we cross-link with formaldehyde, which I think is unsurprising. We've done ultraviolet laser cross-linking instead. We get similar protein occupancy profiles, but we haven't looked as deeply at whether or not we're pulling other proteins out as well.
Excellent. So there are a couple other questions, but they can be answered directly by Peter.
Yeah, and I would invite anyone who's listening and who has a question, please feel free to contact me. I always enjoy discussing science.
And remember that this seminar, the two seminars, are going to be available online on a RNA Collaborative website and YouTube channel. And our next speakers in 2 weeks' time, exactly 2 weeks, Wednesday the 20th of May, will be Morgan Sammons on core stress response and Ellen Chen on simulating RNA folding pathways, hosted by Marlene Belfort on behalf of the Albany RNA Institute. And then